Meiosis is the cellular process to get ready for making babies. It happens in men and women. It happens in plants and animals. It happens in produce and fungi too, but their life cycles are a little weirder. So we'll just stick to pretty much plants and animals, mostly with a human focus, and figure out how this works. It looks a lot like mitosis that we studied a while ago in terms of the process of pulling apart chromosomes. We'll mainly focus on the chromosomes themselves and how those get split apart in the end, rather than the step-by-step process. One of the key things about meiosis to be looking for is that it results in genetic variation. It's the reason that no two people on Earth look alike. And even siblings, even though they share the same parents, have basically no shot at looking exactly like one another. Okay, except for identical twins, but that's a different story. I'm going to tackle meiosis a little bit differently than usual. So these are the topics we'll go over. We'll mostly look at the animation on the screen. And I want to start with a review of life cycles to put this into context, and also chromosomes, because it's been a little while. Let's go back to a super simplified version of an animal life cycle. So up here we have the multicellular adults. They have 2 sets of chromosomes. So we'll refer to that a few times. There's diploid, or 2N. And they, in certain cells of the body, go through meiosis to produce the gametes, the sperm and the eggs. And so gametes is another term you'll hear lots, and that has 1 set of chromosomes. We call that haploid. And those go through fertilization to produce the zygote right here. And that divides through mitosis to produce the chick, which divides more by mitosis to become an adult. Well, here we're only focusing really on the meiosis part of this, which is part of the life cycle. We should note that mitosis is part of a cell cycle, so don't mix up the two. Back to meiosis. Meiosis produces gametes, and this only happens in special reproductive organs. So gametes are just made in plants, in the ovaries and the anthers. In humans and other animals, they're made in the testes or the ovaries. That's really it. But it's all about the chromosomes and who passes on what. And so this one's flipped upside down, so to orient ourselves here, everything on the lower side is diploid, has 2 sets of chromosomes, but they only get to pass on 1 set of chromosomes. That's what haploid means. So the rooster gets to pass on 1 set of chromosomes, the hen passes on 1. That's assured through the process of meiosis. When those come together in fertilization, it makes sure that the chick just has 2. We're focusing on meiosis and how we get it from 2 sets of chromosomes to just 1. Not only just 1, but exactly the right set, so we have 1 of each. Well, you remember that chromosomes come in pairs, and what we have here is a homologous pair of chromosomes. It's going to be important that you remember the difference between homologous chromosomes and sister chromatids. Homologous chromosomes, we got this chromosome from dad, and we got this chromosome from mom. They're similar, they contain the same genetic information, but different versions or different alleles. So that's homologous chromosomes. But sister chromatids, those are made, remember, during interphase, specifically during the synthesis phase. And that happens when DNA replicates. So there's a bunch of concepts from the past that we've looked at, all coming together here, and that produces these identical sister chromatids. And these down here are also sister chromatids. All right, so now we're ready to move on to just an overview of meiosis. You'll find it looks a lot like mitosis, but with some really important changes. Let's start looking at meiosis through that silly one I had before, shoe meiosis. Remember this idea that chromosomes come in pairs, and homologous pairs are kind of like our right and left shoes. And so we'd start that whole process by replicating each shoe, and we'd start being able to move things around, line them up, but this time we line them up differently. right? So here, it looks like we have all the right shoes over here on this side and all the left shoes over here on this side. So that's a little bit different, a really crucial difference. Those get pulled apart, circled back up. Now we've got two cells, and we're going to do that again because we still have those two right shoes together, right? So right in here, I guess that's the left shoe. We have two of those together. We don't need two of them. So we need to separate those as well. Those are the sister shoes or sister chromatids. So we're going to use those microtubules again to push things around, to pull them apart, circle them back up, and there's our final product. Now, notice that everything over here, these are right shoes, and everything over here is left shoes. So unlike mitosis, where we got a closet full of right and left shoes, here we just get 3 left shoes. And we need to combine those with the right shoes of another individual to get a whole set and make a complete individual. Well, let's look at that in a somewhat more formalized way. So here's all the major phases, and I'm going to note that there's a big dividing line. Over here we have meiosis 1 and then meiosis 2. And really, everything interesting happens in meiosis 1, so that's going to be our main focus. But a brief overview, we don't focus quite as much on the phase names, but some critical ones are prophase 1 and metaphase 1, so be looking out for those in particular. So notice here in prophase 1, we're grouping up those chromosomes. Here we have 6 chromosomes, and they are coming together in homologous pairs. So you notice that you got a blue and a red together here and here and here. So that's where the difference between meiosis and mitosis really starts, is homologous pairs come together. And then homologous pairs line up. Then everything else starts looking familiar. They divide, and then they get pulled apart into 2 cells in that telophase. So at this point, once they divide and we enter the second part of meiosis, each cell only has 3 chromosomes, right? 1, 2, 3. So we've already reduced our chromosome number, but we still have those sister chromatids. So we need to divide those up. Otherwise, we're going to end up with double the amount of DNA that we need. So we line them up in metaphase, we pull them apart, put them in different cells, and our result is 4 cells, each with 3 chromosomes. And that's in contrast to the 1 cell we started with 6 chromosomes total. We've cut it in half. That's the main idea. All right, so with that brief overview, let's revisit the important parts. And all of them are in meiosis 1, starting with independent assortment. And that's when, in metaphase 1, those homologous chromosomes line up. Do they line up with the red on the left and the blue on the right, or they switch? And what happens to all the rest of those homologous pairs of chromosomes? Let's find out. So homologous chromosomes line up during metaphase 1. We call this independent assortment. Seen that term before with Mendel. We'll return to that as well. But we're talking about this phase right here when homologous pairs line up. And so when they line up, let's say we had 3 pairs of chromosomes, they could line up in 8 different ways. So here we have all the blue on the left and the red on the right. Here we have exactly the opposite. And in between we have all the different possible combinations. Now, I think it's interesting to go into just a little bit of math here to say, how many combinations could we get? So for this pair of chromosomes, there are 2 ways we could line those up. And for this pair, since we have 2 chromosomes, there are 2 ways we can line those up. And for the third pair, same thing. And so mathematically, that's just 2 times 2 times 2, which is the same thing as saying 2 to the 3rd power. See, it's a good thing you took all that math. Now you get this. And that means 8 possible combinations, which is what we got here. Well, what would that be for humans? how many pairs of chromosomes do we have here? These guys had 3 pairs, but we have more than that, right? And for each of those, we have 2 chromosomes to choose from. We had how many? 23 pairs? What is that? That's almost 8.4 million. Wow. So just by arranging those chromosomes, let's back up to this stage, just by arranging these chromosomes differently, there are 8.4 million combinations of chromosomes that you could come up with. Wow. And this brings us back to Mendel, independent assortment. He talked about this half a century before anybody really could describe it. And so he talked about the law of segregation and independent assortment. And his was based on the observations of these physical characteristics. We could get peas with yellow pods or green pods, but there's also this business of whether they had a smooth coat or were bumpy. And so there were different combinations which sorted themselves out independent of one another. And we now know that's because of the way these chromosomes line up. Is it blue on the left and red on the right? Or is it a different combination of that that results in new combinations of traits?